So take our same, wherever you had your Excel file that had our genes on there, let's find the worst enemies. So again, wherever you have your Excel, let's sort smallest to largest. So I got ESRP2 as the top of my list. The gang that's associated with a gene we just looked at, here's the anti-gang, right? Do you think it's going to have the same functions? Probably not. Well, let's see. All right, so again, let's get the top 200, go down to 201. Control copy. Go back to our browser. Get a new one. Multiple proteins, paste them in. Homo sapiens, search. Yep, continue. This looks a little different. Not quite the same. It almost looks like, is the thing you're studying, that gene you were talking about, is that is it a tumor suppressor?
Yeah, so when that gene is present, there's like tumors are present.
Tumors are or aren't?
They are.
Okay. So when it's expressed, you see tumors.
Yeah.
But when it isn't, it isn't.
Yeah.
You don't see, okay. Huh, interesting. In this case, when I'm looking at this looks like a bunch of, I see CDH1, which is cadherin. This is all about cell adhesion stuff, for sure involved in cancer. But actually, I think it's anti-cancer. PKP3, I've heard of that before. Yeah. So if we look down, let's see what these things do. Again, biological process, not as much connectivity as in the other one. This is all about cell adhesion. Holy crap. Differentiation. Actually, I think this is, your pathway is oncogenic. This thing is trying to prevent that, I bet. Because you've got epithelial cell differentiation, which is the exact opposite of the cancer, right? Making these cells to specialize. You're not despecializing them to cause chaos. But what I find interesting is this cell-to-cell junction. When cells, the thing that prevents cancer is when they come together and they start attaching to each other and they start talking to each other, right? That's a huge thing. So this thing must be involved in breaking that up potentially. And if we look here, a lot of like cell-to-cell junctions. And then what you can do is you can look down here, you can see reference publications, and then basically what it's doing is it's pulling up publications that produce the same genes that you wouldn't expect by random chance. And this top one is study on differently expressed genes in participating pathways of atopic endotermium in adenomyosis patients with different data sets. Huh. So this is probably differentiation, looking at the differentiation of these cells, cadherin. Wow. Definitely associated with cancer if you start reading these papers, but again, this is probably what this is doing. Again, this is the anti-gang. And for me, it does look like that this might be the cops and the other ones are the robbers. So when the robbers arise, they push the cops down, and maybe vice versa. And that's why you're seeing, when you do see the cancer, that's why you see the expression of that gene. And I bet if you used one of the top negatively correlated genes and you have your gene, you could probably, if you took a ratio of that, it would probably give you an indication of how severe the cancer is. Does that make sense? Cool. All right. Anything else down here I need to show? The one thing I will show that's kind of nice to do with these graphs, if you go down to settings, so if we're on the string graph, go to settings, you can do your confidence on what you want with the connections. But here's what I like is I go hide disconnected nodes from the network. It'd be the third one up from the bottom. And you can also, do some kind of like make the gene names bigger if you want it. I could hit update. And now what you're showing is basically the network, but you got rid of everything that doesn't have a connection with something in here. Just out of curiosity, what do you think is the main driving gene in this one? Look at it.
CDH1.
Hell yeah. That is a godfather that is not hiding very well. Totally. I see you. You're guilty. Although it's not a godfather, right? It's probably the captain of the police force. If I wanted to counteract your gene, what I'd probably do is want to induce this one. Does that make sense? Because if I induce this, it's probably going to affect all this stuff. What I want you to see, and what I love these diagrams, is all those stupid pathways you see in the book where it looks like a tube that goes here and you start with this and then you end up with this. It's crap. It's not real. It's drawn by engineers that don't have any idea about biology. Your true pathways probably look more like this, right? The problem is it's very hard to put that on a diagram and go, hey, here's the pathway, right? But again, if you drill down and you figure out what this is, it gives you an idea. You can figure it out. And I will say, this is probably the first time anybody has seen this pathway. The great thing about bioinformatics is every day I discover things that no one else in the history of the world has ever seen. Do people not believe me sometimes? Hell yeah. That's the blowback you're going to get from all of this stuff, is people go, well, you didn't do any experiments. How do you know? Because I analyzed about $1 million worth of data and I came up with this. Does that make sense?